Okay, let's unpack this a bit. Imagine your body as this incredibly complex machine, right? All these different parts working together.
Hmm.
Now, what happens when one maybe seemingly small part starts acting up? You might think, okay, local problem.
Right, you expect it to stay contained.
Exactly. But sometimes that single glitch sends like shockwaves through the whole system,
Yeah.
affecting things you wouldn't expect.
And that's really the core of inter-organ communication, this crosstalk. It's fascinating and sometimes, yeah, a bit concerning.
It really is. And the specific crosstalk we're diving into today might sound, well, a bit odd initially. We're talking about your kidneys and your lungs.
Uh-huh. Specifically, what happens in the lungs when the kidneys suddenly run into trouble, what doctors call acute kidney injury, or AKI.
Right. And it's not just the obvious thing, like, oh, the kidneys aren't clearing fluid, so the lungs get waterlogged. This is something else.
It is. Recent science is pointing towards a much faster, more direct link, a kind of rapid response.
Yeah, we're not just looking at two things happening around the same time. The research we've looked at, especially this commentary on a really interesting new study, digs into the actual mechanics of how this kidney-lung talk
The
happens.
nuts and bolts of it.
Exactly. So our mission today is to understand how AKI can almost immediately mess with lung function right down at the cell level, potentially making it hard to breathe, even without that classic sign of fluid overload. It's surprisingly quick.
Okay, so let's set the scene. AKI, acute kidney injury, it's actually a really common issue, especially in patients who are critically ill.
Yeah, you hear about it a lot in ICUs.
You do. And when the kidneys get hit suddenly like that, it's not just about filtering waste. It kicks off this whole cascade of events, including significant inflammation throughout the body, a systemic response.
And that body-wide inflammation, unfortunately, doesn't just bypass the lungs, does it?
No, not at all. We've known for a while that AKI is linked to worse outcomes overall, and that includes injury to the lungs.
But the frustrating part, I gather, has been pinning down this specific kind of lung injury. Like, how do you diagnose it? How do you treat it when it stems from the kidneys?
That's been the challenge. And one of the really intriguing observations clinically is that patients with AKI can develop hypoxia, low oxygen levels, even when they don't seem overloaded with fluid.
Right. Their lungs don't sound wet, maybe their x-rays aren't showing massive fluid.
Exactly. And even measures like the pulmonary artery wedge pressure, which helps to check for fluid back up from the heart into the lungs, can be normal. That strongly suggested something else was going on.
And some clues started turning up in autopsy studies, didn't they?
They did. Pathologists noticed things like protein-rich fluid, these things called hyaline membranes, and significantly, a lot of white blood cells, leukocytes, infiltrating the lung tissue in patients who'd had AKI.
Which led to that older term, uremic pneumonitis.
That's right. Though maybe a bit of a misnomer in some ways, but it pointed towards this kidney-lung connection.
The problem, though, is that this often happens when patients are already really sick with other things like ARDS.
Acute respiratory distress syndrome, yeah. Or they do also have fluid overload. So picking apart the specific contribution of the AKI itself to the lung problem is really tough.
It muddies the waters.
Completely. It's
Wow.
hard to see it as its own distinct thing sometimes.
And it's also quite striking you mentioned that AKI often leads to worse outcomes than, say, chronic end-stage kidney failure.
It does seem that way,
Mhm.
which suggests that the acute injury, the suddenness of it, triggers some particularly damaging changes rapidly.
And there was a study finding, I think, where mechanical ventilation was the only thing boosting mortality risk in AKI patients.
Yes, in some studies, needing a ventilator because of respiratory failure was a key predictor of worse outcomes in AKI.
Yeah.
That really underscores this tight link between kidney failure and lung dysfunction.
Okay, so this brings us to the new research trying to shine a light on this. Kamaru and colleagues, right? Using a pretty advanced technique.
Yes, intravital microscopy in a mouse model of AKI. It's incredible. It lets you actually watch biological processes happening in real time within a living animal's tissues.
So, peering right into the lung capillaries, what did they actually see?
Well, the speed was the first striking thing. Within literally 5 to 10 minutes of inducing AKI in these mice.
Wow, minutes.
Minutes. They saw this dramatic accumulation of neutrophils, a type of white blood cell, sort of the immune system's first responders, right there in the lung capillaries.
Okay, neutrophils flooding into the lung vessels. Were they the only ones?
They noticed they were actually preceded by monocytes, another kind of immune cell. The monocytes seemed to arrive just ahead of the neutrophils.
Okay, monocytes, then neutrophils. But here's the really crucial part, as I understood it. where did they go? Did they, like, burst out into the lung tissue?
No, and that's the key finding. They migrated to the lung capillaries, but they overwhelmingly stayed inside the blood vessels. They didn't extravasate, didn't move out into the surrounding lung tissue, the alveoli.
So they're just piling up within the pipes, essentially.
Exactly. The researchers described seeing these neutrophil trains almost like a logjam. And it looked like these were being caused by slow-moving monocytes, specifically ones carrying a marker called CCR2.
CCR2 positive monocytes acting like, what, traffic cops slowing everything down?
Sort of like lead cars in a traffic jam, maybe?
Yeah.
And these jams of neutrophils physically slowed down the blood flow through those lung capillaries.
Okay, that sounds like it would definitely interfere with gas exchange.
Absolutely. And when the researchers disrupted this process, interfered with this pileup,
Oxygen levels improved.
oxygenation got better in the mice. It's a pretty compelling link.
So it's less about leaky vessels flooding the lungs, at least initially, and more about this cellular gridlock within the vessels themselves.
That seems to be the primary mechanism they identified in this very early phase. They contrasted it directly with what happens if you induce lung injury using something like LPS, a bacterial component.
What happens then?
In that case, the neutrophils do leave the blood vessels. Something like 80% of them moved out into the bronchoalveolar space, the air sacs. So a totally different response pattern.
Interesting. And in the AKI mice, when did the breathing problems actually show up?
They could detect hypoxemia, the low oxygen, around 2 to 6 hours after the initial kidney injury.
Okay. And what if they, say, got rid of the neutrophils? Did that help?
Yes. When they depleted the neutrophils, blood flow in those capillaries was restored and oxygenation improved. It really points the finger at that physical pooling of neutrophils as a direct cause of impaired oxygen exchange.
Did the neutrophils themselves change after the AKI? Did they look different?
They did notice some changes. The neutrophils appeared more rounded, and they had a stronger signal for something called F-actin.
F-actin. That's involved in cell ship and movement.
Exactly. Researchers suggested this might mean the neutrophils were actually less stiff.
Less stiff. Wouldn't that make it easier for them to squeeze out?
You'd think so. But the idea here is maybe they become too deformable. Or perhaps their ability to interact with the vessel wall to find an exit point is impaired. It suggested a reason why they might be getting stuck inside.
But they weren't, like, paralyzed. They could still move if called.
That's right. If the researchers put a chemical signal that attracts neutrophils, a chemotactin called CXCL2, directly into the mouse's airway,
the neutrophils could get there.
they could still migrate out into the air spaces. So the cells could move, but in the context of AKI, something was keeping them within the vasculature.
Okay. And you mentioned monocytes arriving first. Did it matter which type of monocyte?
Yes, this was another really critical finding. They found that depleting classical monocytes specifically,
Not other immune cells like alveolar macrophages, the ones already living in the lungs.
right, not the resident macrophages or the non-classical monocytes. Depleting just the classical monocytes stopped the neutrophils from piling up in the lungs.
Wow. So these classical monocytes seem to be the key orchestrators of this neutrophil traffic jam.
It strongly points to them. It suggests a very specific cell-to-cell communication pathway kicking off this whole response.
Now, we should probably acknowledge what we already knew or thought we knew about AKI and lungs. There's the whole issue of edema, right? Fluid buildup.
Absolutely. Previous work, both in humans and animal models, has definitely shown that AKI can lead to lung edema, leaky blood vessels, that endothelial barrier dysfunction. And that certainly can impair oxygenation.
But the Camara study, they didn't find much evidence of that edema, did they?
Not by their standard assessments, no. They didn't report significant edema in the tissue histology. It raises questions about the relative contribution of these neutrophil trains versus the more traditional edema, especially under the conditions they use, like high oxygen levels during ventilation.
So the jury might still be out on how much each contributes.
Or perhaps they contribute differently at different time points or under different severities of AKI. It's complex.
Were there any other culprits? Did they see anything else that might hinder gas exchange?
They did note evidence of microthrombosis, tiny blood clots forming in those lung capillaries after AKI, based on specific staining.
Ah, so clots could also be gumming up the works.
Potentially, yes.
Yeah.
That's another factor that could definitely contribute to problems with oxygen and CO2 exchange.
Okay, so we have physical blockages, neutrophils, maybe clots, and maybe some leakiness. What about the signals, the molecules triggering all this? We know AKI sparks inflammation.
Right. We know cytokines and various immunoregulatory molecules get released. And interestingly, some experiments where they actually remove the kidneys, a nephrectomy.
A lung injury still happens.
In some models, yes, which strongly suggests the injured kidneys are actively releasing something that travels to the lungs and causes trouble, rather than the lungs suffering just because the kidneys aren't filtering.
So those uremic factors, the waste products that build up, are they the messengers?
They might play a role in the broader picture, especially longer term. But for this very rapid leukocyte accumulation happening within minutes, uremic factors probably aren't building up fast enough to explain that initial wave. Something more immediate seems to be released by the injured kidney.
Okay. And we know some inflammatory signals, like IL-6, can draw neutrophils to the lungs via another molecule, CXCL1.
Yes, that link's been shown. But IL-6 alone doesn't seem to be sufficient to ramp up CXCL1 production enough, suggesting other factors are needed to really get those neutrophils moving en masse to the lungs.
Which brings us to osteopontin.
Exactly. This is work from another group, the Herlick lab. They identified osteopontin, a protein released by the kidney cells after AKI, as a key mediator driving the subsequent lung injury, the endothelial leakage, the inflammation, the respiratory failure.
So the plot thickens. Could osteopontin, or maybe IL-6, be the signal that tells those classical monocytes we talked about, the ones already in lung capillaries, to start pumping out CXCL2?
That's the big question connecting these findings. Because the Camaro study really zeroed in on CXCL2 being the critical attractant for the neutrophils within the lung vasculature released by those classical monocytes.
And they confirm this how? Through single-cell analysis.
Yes, they use single-cell RNA sequencing. It lets you see which genes are active and therefore which signals are being sent and received by individual cells. Their analysis pointed strongly towards this CXCL2 communication happening between classical monocytes and neutrophils, right there inside the capillary lumen.
And crucially, the resident lung immune cells, the alveolar macrophages, they weren't doing this.
No. Unlike in direct lung injury where they'd get activated and pump out inflammatory signals, after AKI, these resident macrophages didn't show that kind of pro-inflammatory shift or CXCL2 expression. It really reinforces the idea that this is an intravascular event driven by circulating cells.
So this all paints a fascinating, if slightly alarming, picture. What are the broader implications? What did Camaro and colleagues speculate about why this might be happening?
Well, they floated a really interesting idea. Maybe this trapping of neutrophils in the lung capillaries is actually a kind of protective mechanism gone slightly wrong.
Protective? How could blocking lung capillaries be protective?
Their thought is, perhaps it's a way for the body to quickly contain a massive wave of activated neutrophils triggered by the AKI, preventing them from circulating widely and causing damage in other organs, sort of like creating a specific sink for these inflammatory cells in the lungs.
So the body might be trying to sacrifice local lung functions slightly to prevent a systemic inflammatory meltdown, like an emergency brake.
That's the speculation, a potentially costly trade-off, where containing the fire locally impairs oxygen exchange. It's a provocative thought.
Definitely. So if that's the case, how might we intervene?
Well, one idea they propose is directly targeting these neutrophil trains, finding ways to break them up or prevent them from forming.
Okay, treat the traffic jam itself.
Right. But perhaps a more upstream approach would be to target the signals that cause this whole intravascular chemotaxis in the first place. Block the signals telling the monocytes and neutrophils to gather there.
So focusing future research on things like CXCL2 or osteopontin as potential therapeutic targets.
Exactly. Understanding the compartment-specific roles, what these molecules do inside the vessel versus outside, could be key to developing therapies that prevent AKI-induced respiratory failure without, say, broadly suppressing the immune system.
There's still clearly a lot to figure out, though, the interplay between these neutrophil trains, any edema that does form, maybe those microclots.
Absolutely. It's likely a combination of factors contributing to the hypoxemia we see clinically. Untangling that complex interplay is vital for developing better, more targeted treatments and improving how patients fare after AKI.
Okay, so let's try and boil this down for everyone listening. The big takeaway here seems to be this surprisingly fast development of a sort of cellular traffic jam in your lungs' smallest blood vessels right after an acute kidney injury.
Driven by neutrophils piling up.
Right. These neutrophils, guided there by classical monocytes, releasing signals like CXCL2, and this jam can directly interfere with your lungs getting oxygen into your blood, even if your lungs aren't full of fluid in the way we typically think about.
It really highlights just how interconnected everything is.
Yeah.
What happens in the kidneys can have these immediate, quite dramatic effects elsewhere, like the lungs.
It does. It's a powerful reminder of that intricate organ crosstalk.
And you have to wonder, this very specific rapid cellular interaction they've uncovered in the lung vasculature after kidney injury, could understanding that unlock new ways to treat respiratory problems, not just from AKI, but maybe other conditions involving widespread inflammation. It makes you think differently about how a problem that seems localized to one organ can trigger such specific microscopic chaos in another, doesn't it? A lot left to explore there.